Hello and welcome. This is Biology Practical 1. So I want to, if you go on net now, you will see for Neco exam, Nigeria, you will see the specimen is already there. Many students have access to it already. So in specimen A, what you have there is what? Is onion bulb. Specimen B, you have ginger, rhizome. Specimen C, you have Irish potato, that's tuber, right? So specimen D, you have a potted bean seedling, 1 week old, right? Specimen E, we have potted guinea corn seedling, 1 week old. Specimen F, you have head of a pigeon with neck. So specimen G, you have the atlas vertebra of a rabbit, and specimen H is the lumbar vertebra of a rabbit, which is what we have here. Now, the purpose of this lesson, the main reason I decided to teach you now, is to guide you on how to draw and label, and as well give you some important information on the relationship between the specimens. So we have look at this diagram now. I want to guide you on how to draw. If you have been given, we have discovered that after teaching students, a student will be asked to draw. You see something that is something like this. The student to draw it very small. You now wonder, it's not even to the size. You now wonder, how come? Very small. Or sometimes you see a shading. They will draw something and shade. Even when they clean, it is not. They don't clean it completely. So I'm here this moment to guide you properly on how to draw and draw well. Now, the first thing you do while drawing is to ensure that you draw with one free hand. Free hand. You don't need to shade. When you are drawing something like this, look at this. You have something like this. Can you see this? How you are supposed to hold your pencil? Can you see it now? So it's very simple. It's very simple. It's in form. Something like this. It's very simple. Then you put this. Put this. Though, because I've learned how to draw it anyway. Right? So it's a very simple drawing. You don't need to start shading and start doing other things. It's wrong. So some students will draw. When they draw with, let me use the word muzzle. Then after drawing, to clean becomes a problem. After cleaning, you see traces of the already drawn diagram. So if I clean something and it still appears like this, it means it may look like part of the diagram. So it's not a neatly labeled or neatly drawn diagram. So it's not neatly drawn. Please. Now this is for use free hand. When you want to erase completely. Don't draw it to make so much mark. I hope you can understand. And avoid what? There should be no shading. Don't shade any part unless I can't remember the time it's required, but you don't do shade. You don't shade anything. But sometimes some diagrams can have dots. It's part of the nature of the diagram. Did you get that? So, but there should be no shading of any kind. This one is also supposed to be like this here. So there should be no form of shading. Please don't shade anything. Now, when you draw to what? Size. You draw to size. Sometimes you see students, this is the paper. They will just draw something like this. Let me just say, like plant cell. They will just draw something like this, very small like this. You now start wondering why the size is given there. You may be asked to draw a diagram 8 cm to 10 cm long. How do you approach this? 8 cm to 10 cm. You look at what you have to do. You get your ruler, something like this. If I want to draw it 8 to 10 cm long, I will get my ruler, measure like this, possibly 10 here to here, or 8 from here to here. I will measure 8 or 9 within the range. I measure here 10 like this. It's still within this thing. I will just make a mark here with pencil dot. Make another dot. Make another dot. Make another dot. I will now draw it within this size, so that at least the size will be enough. So if you draw it less than the size, you are going to lose some marks. So make efforts to look at the size and draw it to that very size. I hope you can understand that. Very, very nice. This is the mistake many students have made in the past, and I don't want you to repeat such mistake. Please, that is the first thing that the person that is going to mark your script will look at. The size. Look at the size. Then the next thing I want to tell you about is how you label You are drawing. Labeling. You are labeling. This matters a lot. You are labeling. How do you label? This is another important thing. Can you observe some things here? Look at this. The first thing, this is not so common anyway. Capital letter N, capital letter S, capital letter P, M, capital letter N, A, N, C, T, P, C. So this is how you are supposed to label it. Use title case. Start with capital letter. Some persons will label this thing with capital letters all through. Capital N, capital E, capital letters all through. That is not bad. I hope you can observe that. So don't start. Students have seen that. Some students will start with small letter. So it is better. Quote me. It is better to start with capital letter. Each of these, make it title case. It is, quote me, better. All right. So now look at this. Now look at, if you want to label, sometimes some students hastily, this place is not the same as this place. Watch this point. This here now is not the same as here. They are different parts of this. See, this place is different from here. But you will see students after drawing it on the board, you ask them to draw in the classroom, they will give you a different thing. I don't know. Something that is here, they will just extend it. I keep wondering, what is the problem? So you cannot see, or are you blind? I keep wondering. How you can observe that? So let it be precise. Be precise in what you are labeling. That place you are supposed to, like this place that is here, you don't need to put it here. This one is inside. Put it like this, as it is. All right. So now look at another thing I want you to observe. You draw the lines using these lines. Use ruler. Use ruler. Draw these lines. I've seen many students hastily. They use free hand like this. It's wrong. Please, you don't need to do that. Use your ruler. Draw it with pencil like this. Like this. Rule it very well. Very neat. Rule it very neat. So now you must avoid crossing lines. Crossing lines. If you have something like this, let me say something like this. You are labeling this plant cell like this, like this. Then the person does this one. Remember, right? Draws this one. Now remember, comes and draws this one like this. This is wrong. You don't cross lines because maybe you don't have space here. You now decide to bring it here. You don't cross lines. When you cross lines, it's wrong. So, and it is better to have all your labels at one side. Look at it. All of them is better. But some diagrams, like the grasshopper drawn. Let me just see how I can manage it like this, like this, like this, something like this. Then you give it legs. No one to give it legs. Now, give it legs. Something like this. So if there's something like this grasshopper, you label it. You can label it like this, like this. Then the back, like this, like this. So you can label it in this way. This one like this like this. If you know you are going to find it difficult to put all of them at one side, but if you look at it, it looks neat. Something like the mammalian heart, that has different parts. It may be somehow difficult to label it one side. Maybe somehow difficult to label it and go one side. Something like this. So it may be somehow difficult. The mammalian heart, it may be somehow difficult. So you can label it left, right ventricle, left auricle, the septum, the outer, and the rest of them. You may put them the two sides because it may be difficult to put all the parts at one side. I hope you are getting it now. So please, when you are labeling, you should be very, very careful. This is what will give you the mark you desire or you want to get. So I would like you to learn this. Learn this. This is neural spine. This is hypophysis. This is metapophysis. This is neural arc. This is neural arc. This is neural canal. This is transverse process. And this is the centrum. This is the anterior view of specimen H. What is specimen H? The lumbar vertebra of a rabbit. So we are going to now look at other diagrams. So let me look at it. Specimen F, the head of a pigeon. Let's label this. Remember, always put the title. Drawing on specimen F. right? So this is the head of a pigeon with neck. So you label one side. This is the head. This is the eye. This place is the what? The cherry. This is the nostril. This is the beak. And this is the neck. So it has been labeled. Now, there is something, okay, the way you may be asked to draw this, but you will be asked to specify. Know that the kingdom, what is the kingdom of this bird? Let me add information. What is the kingdom? You can say kingdom what? Animalia. Kingdom Animalia. Then, what is the phylum? Phylum. It's a chordata, because it starts backward. It's a chordata. Then class. Under class is under aves. Aves, that is birds. Kingdom Animalia. Animalia, phylum chordata, and class aves. So you may be given this question. State the kingdom, the phylum, and class of specimen F. Then I want to ask you another question that you may be asked here is what? The relationship between F and which one is the seedling? Okay, the seed of specimen E. Yes, guinea corn. So this bird feeds on guinea corn. Guinea corn is a concentrate. Corns are called what? Concentrates. Birds take corns to give them energy, concentrates. So it gives them more energy. That energy is for flights. So they take concentrates so that they will have energy to what? To do what? To fly. That's the relationship between specimen E and F. Now we are going to look at another diagram. So let's look at which one? The onion bulb. All right. Okay. This is okay, let's label this drawing. This is the longitudinal section. That is LS of specimen A. Yes, specimen A. That is the onion bulb. All right. So let's label this. This is a bulb apex. This place is the membranous leaf. This place is the fleshy. This is the membrane. That one you remove is the membranous leaf. So this one is the fleshy. Please remember to use ruler. So fleshy leaf. This place is the flower bud. Then this one is the lateral. Let's use capital letter. You can still use L, but I prefer this. Lateral bud. Then this is the stem. And this place is the adventitious. Okay, adventitious roots. So let's do it like this. Adventitious roots. So can you see it? That's good. Please learn this. You can do it. It's not difficult. You've seen that I've been able to do it, so you too can do it. So it's not a difficult thing. So now what I want you to do is to go and do some kind of the types of roots, of the specimen A, B, and C. How they grow, their relationship, and their uses, the functions of the parts. So you need to know that one. So let me give you the relationship between specimens D and E. I know that one is very, very necessary. The two, the beans. The bean is a dicotyledonous plant, while the corn is a monocot. Bean is dicot. This one is a monocot. A week old of the seedling. So it's just like a young plant, the one that undergoes apogee and hypogee germination. So please go and learn it. But what I want to do now is to give you in tabular form the relationship between the similarities and differences between what? Specimens D and E. that is a bean seedling, and guinea corn. Alright.